Hi, BISC 210 students. Today we're going to be covering Chapter 8, Microbial Metabolism. Now, this is going to be quite a bit of review for you all. We've talked about many of these concepts already in terms of anabolism, catabolism, enzymatic activity, but then we'll talk a little bit about some methods of exchanging genetic material. So there will be some material, again, that just looks familiar, and hopefully you are familiar with it from previous lectures, but some will be new as well. So first off, the term metabolism. When humans hear the term metabolism, most of us automatically think of digestion. It just goes, that kind of goes hand in hand. People say metabolism, they say, oh, digestion. But metabolism is actually a broader term than that. Metabolism actually refers to all chemical reactions in a cell. Now in humans, that includes digestion. But in bacteria, any reaction that occurs falls under this umbrella term. Same thing in humans, falls under the term metabolism, all chemical reactions. We just happen to associate one specific chemical reaction with this term as humans. But it actually means all reactions. Now, there are two major pathways, kind of what the book calls yin and yang, basically two reactions that occur where one does one job and the other does the exact opposite. The catabolic and the anabolic pathways. Catabolic essentially is breaking down substances to release energy. Now, if you recall from earlier in the semester, I said that there are several places we store, we as well as bacteria store potential energy. And one of those places is chemical bonds. So within chemical bonds, there's a lot of potential energy. And if you break those bonds via catabolism, then you're going to release that potential energy, and it'll be utilized to power some reaction doing something somewhere. Anabolic pathways, on the other hand, are energy-using pathways where we're building new substances, creating chemical bonds, which costs energy. So catabolic releases energy because we break the bonds, and then anabolic is we use energy because we're building bonds, or building molecules. So it's important to know the difference. Now with each one, we talked about a specific reaction. So for instance, with catabolic reactions, we talked about hydrolysis. Hydro meaning water, lysis meaning break. So in hydrolysis, let's say we have a complex sugar, and the cell wants to break down the sugar into smaller sugars, monomers like monosaccharides, to get some of the energy out of there. So cells use hydrolysis, a water molecule or multiple water molecules, to break the chemical bonds to release the potential energy that's stored within that particular reaction. Then we actually have the complete opposite of that, which is the anabolic reaction, or the dehydration synthesis reactions. Now, if you recall, dehydration synthesis reaction simply means dehydration, well, that means we're losing a water, and then synthesis means to build or to create. So we're losing a water molecule to create chemical bonds, to build from monomers to polymers. And when we build those bonds, we have to spend energy. Same thing with bacteria. They have to spend energy to do this. So anabolic reactions require the input of energy to create chemical bonds and build larger polymers, where catabolic break the bonds and release the energy. And this is this is really common, this image. I really like this image. It just shows you that catabolic and anabolic reactions are what we call coupled, meaning you have one happening and the other one will happen as well. So for every catabolic reaction, you're going to have an anabolic. Catabolic and then anabolic. Now, not every single time, but pretty close. So you're going to break something, build something, break something, build something, over and over and over again. We're going to take this, let's say this is a polysaccharide. We're going to break it down into simple molecules, release the energy, use the energy, and then when we have this, now we want to build it back up. We're going to use an anabolic reaction, dehydration synthesis, which requires energy to get a more complex molecule. So it's important we understand that these reactions are typically coupled, meaning they happen together. ATP, adenosine triphosphate. If you've taken any biology course prior to this one, you probably know what ATP is. Adenosine triphosphate is the energy storage molecule that will provide the energy for anabolic reactions. So it's made by catabolic reactions and then provides the which provides the energy for anabolic reactions. Now ATP is used in humans as well. Our cells use ATP. Specifically, the mitochondria produce ATP within our bodies. That's one thing that is focused on in high school biology, that the mitochondria is the powerhouse of the cell, meaning it makes tons of ATP. Well, there's truth to that. The mitochondria in human cells make lots of ATP. But we know from this class that mitochondria actually used to be their own organisms, right? Billions of years ago, they were individual bacterial prokaryotic cells. So hopefully you remember that and remember the endosymbiont theory. But ATP is really good at storing a lot of energy to be used later. I'm going to show you the structure of ATP and explain why. So this is what ATP looks like. So you have a very specific structure. You have this whole thing is ATP, adenosine triphosphate. Here's the adenine attached to a sugar, and then tri, which is three phosphates. So when you look at the sugar and the adenine together, we call that the adenosine region. So sugar, the ribose, and the adenine, this is the adenosine region. Triphosphate attached to the sugar, 1, 2, 3 phosphates. Okay. So that's ATP. Now, this image shows you that you can remove one of the phosphates and have adenosine diphosphate, ADP, which is pretty common. And then you can even have adenosine monophosphate, where you got rid of both this one and this phosphate, and you're left with only one of them. So adenosine monophosphate. AMP, we're not really going to talk about. Adenosine monophosphate, yes, is very important, but we're just going to focus on ATP and ADP and how cells constantly take ATP, break it down to ADP, build it back up to ATP, break it back down to ADP, so on and so forth. Now, all chemical bonds store potential energy. Not equally, some store more than others, but they do all store some form of potential energy. For instance, a single bond would store less energy than a double bond. Triple bond would store more potential energy than a double bond. And then there are these bonds between the phosphates, these two red ones. So here's the deal. These phosphates hate each other. They can't stand each other. They do not want to be near each other. They don't like it. Notice there's a negative charge. It's right here. So all of these have a negative charge, all three of them. And what happens if you have like charges? They repel, right? So these three phosphates really hate each other even though they're stuck together. So when they get the chance to break one of these phosphates, this last one off, break this bond right here, they're really happy. The bonds between phosphates are high energy, meaning they have a ton of potential energy stored in them. More than triple bonds, more than double bonds, right? They have lots and lots and lots of potential energy stored within these bonds between the phosphate groups. And you can see the charges down here, negative, negative, negative. They don't want to be near each other, right? All these same charges repel one another. But the molecule is forced to be in close contact with each other. So you have these high energy bonds right here. Oops. So let's say we have a bacterial cell that has something inside of it that it needs to remove from the cell. It's a waste product. Doesn't really matter what it is. It just can't leave on its own. It needs help of a transport protein. So transport proteins, if you're looking at active transport going against the gradient, one of the requirements is energy. So what would happen is an ATP molecule would be close to this this transport protein, and then ATP would undergo hydrolysis, hydro-water lysis break. So a water molecule would break this high energy bond right here. When this bond is broken, several things happen. First off, this phosphate group right here, this thing right here, flies away because it now, because it's repelled, it can be pushed away, and it does. It just floats away. We call that an inorganic phosphate that floats away. This bond that is broken releases a ton of energy to be used to power the movement of the substance via active transport through the membrane protein. And what you're left with is adenosine diphosphate. You have two phosphates and then the adenosine region. So you started with ATP, hydrolysis occurred, breaking this bond, and what your reaction leaves you with is a phosphate that floats away. This one runs away. You're left with ADP, adenosine diphosphate, and then a bunch of potential energy that has been released. Now, potential energy, when it starts to be used, becomes kinetic energy, right? Potential is stored, kinetic is utilized. So it's released. This energy is now going to power the movement of substances across the membrane through the transport protein. Okay. So basically, this is just the ADP and ATP cycling. Once you're left with ADP, the cell can then take one of these phosphates that's floating away, and it can jam it back onto the end of the ADP molecule, recreating ATP. So again, it's cyclical, right? It's one big cycle. You have ATP broken down into ADP, built back up into ATP, broken down back into ADP, so on and so forth, and the energy is released and used as necessary. Okay. Enzymes. So enzymes we've talked about a bit. Right now we're just going to kind of go into them a little bit more detail because they are super important. When we get to later in the semester, we're going to see how bacteria, specific bacteria, use enzymes to damage the host. So they do play an important role. So it's important we understand them. So enzyme has a couple sites to it. One of them is the active site. That's where the substrate will bind. So whatever the enzyme is going to act on or mess with, so to speak, will bind to the active site. Will bind to the active site. Then from there, from the active site, okay, the substrate that's now attached to the enzyme substrate complex, the enzyme does its job. In this case, it's cleaving the substrate in half, and then you get your products. And then the enzyme is now available again to do it again with another substrate. Excuse me. Enzymes are pretty specific, so the active site fits only specific substrates. And it doesn't always have to break things down. It can build things. So if we had, instead of these being the products, those were our substrates. So if we swapped this and these two and made these our substrates and this our product, that's perfectly acceptable. The enzyme would bond them together, link them together, and then you'd get this as your product. So enzymes aren't always breaking things down. They can build stuff as well. So this is just talking about the enzyme binding to the active site. When it binds, it's called the enzyme substrate complex. So this is the enzyme substrate complex right here. And then the active site is where the substrate is binding. This should not be new. This should be a repeat of some of the stuff that we've seen earlier. Now this is important. First off, with enzymatic name or enzyme names, if it ends in ACE, A-S-E, it's usually an enzyme, which are a type of protein. So helicase, gyrase, polymerase, phosphofructokinase, right? All these things that ends in ACE are almost always enzymes. Not always, but pretty close. And then with this is showing us how enzymes speed up chemical reactions. Yes, we've talked about this before, but this image should be new. So basically, when you have a reaction that needs to occur, it requires X amount of energy to start the reaction. Right? So this gray line is saying we need this much energy to start the reaction. Okay, right here. From here to here. We need that much energy to start the reaction without an enzyme. But if all of a sudden we throw in an enzyme or enzymes, notice the amount of energy required to start the reaction significantly decreases. Okay, so it significantly decreases. The point is, enzymes speed up chemical reactions by lowering the amount of energy required to start that reaction. Let me repeat that. Enzymes speed up chemical reactions by lowering what we call the activation energy, the amount of energy required to start a reaction. Notice the gray line, we need all this energy to start the reaction without an enzyme. Toss in an enzyme, now we need significantly less. So that's how we as well as bacteria speed up chemical reactions, by adding an enzyme. And it lowers the activation energy. Again, this is something we've discussed, but I want to make sure that we're clear on it. The question, what would graph look like if enzymes did not increase the reaction rate? So if the enzyme didn't really do anything, it would go back to what it looked like with the gray line, right? the whole point of adding an enzyme is to decrease the activation. Cofactors. So as I mentioned in the previous slide, or 2 slides ago, there's an active site. There's a couple other sites as well, one of them being what we call the allosteric site. And some enzymes won't function unless they have cofactors, such as iron, zinc, things like that. They remain in an inactive form. So when you are looking at an enzyme, if you were looking at the protein portion, so this is just the enzyme itself, and it's not active, this is what we call the apoenzyme. The apoenzyme. Then, if we take the apoenzyme, add the cofactor as well as the substrate to it, this whole thing is called the holoenzyme. It is the active form of an enzyme. And when we have the cofactor and the substrate, you get your products, whatever they are. If you're missing the cofactor, then the substrate may not work, or the enzyme may not work on the substrate, and then you don't have products. So this down here in this bottom portion will be the apoenzyme, the inactive form, right? It's still inactive here, so it's still the apoenzyme plus the substrate. But up here, you have your apoenzyme plus your substrate plus your cofactor, which gives you your holoenzyme, which is the functional enzyme. Now, cofactors can be many different things, but some enzymes absolutely require a cofactor. Not all, but quite a few. Again, in biology, you can't say everything follows a specific set of rules. It's just not the way it works. There are almost always exceptions to the rules or to the concepts. There are almost always, if not always, examples that kind of contradict what we say. But anyway, moving on. Lots of things affect enzymatic activity. One of them is temperature. We talked about this before. Lowering the temperature does one thing versus increasing the temperature. So if you lower the temperature, the efficacy, the efficiency, and how effective this enzyme is becomes significantly less. So it'll work if you lower the temperature, but it won't work efficiently, and it won't be working to its maximum capabilities. If you start to warm the temperature up, though, as temperature goes up and up and up and up, the enzymatic activity increases until you reach the optimal temperature. When you reach the optimal temperature, then it's going to be functioning as efficiently as possible. For instance, in humans, you have to think about our body temperature, 98.6 degrees roughly-ish. Our enzymes have to have an optimal temperature close to that. So our enzymes in our body have to have an optimal range around 98.6. Doesn't have to be perfect, but it should be pretty close. Same thing with bacteria. They should have optimal ranges at which they function. A lot of the bacteria we look at in class, like E. coli, for instance, work really well at what we call 37 centigrade or Celsius, which is pretty much body temperature, a little bit above body temperature. So they hit their optimal range and they're working really efficiently and fast. But if you stray too far above that temperature, it creates problems. If you go too far above, then what's going to happen is the temperature gets too hot, and it will start to denature proteins. Denature basically is destroying. It unwinds them. Proteins have a very specific shape, i.e. enzymes, which are a type of protein, have very specific shapes. So if you denature them by increasing the temperature too much, they become useless. And that enzyme will just be non-functional. It will be non-functional. This is a bad thing. So, in humans, it's kind of interesting. When we get a cut, let's say on your forearm, okay, you notice site-specific inflammation, meaning it's red, it's warm to the touch, it's probably raised, right? It might leak a little with some lymphatic fluid. But you notice that if you touch it, it's warm, right? We call that site-specific inflammation. And basically, inflammation is a couple things that are happening, allowing white blood cells to show up, dilating vessels. But the other thing is increasing the temperature. Because if you can increase the temperature where the bacteria are infecting, and if you can increase it enough, we can theoretically denature their proteins while our proteins are still perfectly fine. Because our proteins have an optimal range, a little bit around body temperature and a little bit above and below. But bacteria, some of them have ranges that are significantly lower than body temperature. So if you get a cut and if there's a slight infection, it can be warm to the touch because our body is saying, well, hey, let me increase the temperature to allow for more white blood cells to show up. Also, let me increase the temperature to try and denature bacterial proteins or enzymes. And then they become non-functional. And then if those enzymes are essential, then the bacterial cell will die. So it's pretty neat how our body responds to that. We'll talk a little bit more about that later on. Here's a great example, just talking about optimal temperature. Notice around 40 degrees Celsius is where the bacterial enzyme is functional. There's its peak right there. And then if you stray too far, notice it drops down to 0. The reaction rate goes down to 0 because, well, there's no more enzymes that are working. If you go lower, it's just less efficient. If it goes too high, it's going to break the protein apart. Now, hopefully you remember what that actually means. Hopefully you remember that hydrogen bonds hold proteins together, and if hydrogen bonds are broken, then protein cannot maintain its shape, therefore it denatures, unwinds, becomes useless. Now, my question to you would be, do you expect every organism, every bacteria, to have the same optimal temperature? Hopefully you answered no. Right? Depending upon the organism, the optimal temperature range will vary greatly. For instance, thermophiles versus mesophiles. Mesophiles are roughly body temperature. Thermophiles are hot at a very high temperature. So there are significant differences between organisms. So looking at microorganism versus microorganism, different bacterial species versus different bacterial species, they may have different optimal temperatures. Now, this is just an image showing you what how denaturation happens. The left is the active protein. It's folded. It's in the right shape that it needs to be in. All of a sudden, the temperature gets too hot, and boom, it inactivates, it unwinds, and it becomes useless. It becomes denatured. pH is the same thing. Enzymes work at an optimal pH. Too high, it will denature as well. Again, I'm really not going to go much more into this because it's the same concept as temperature. pH will alter enzyme structure and will disrupt bonds. You could also say the same thing for salt concentration. You could say the same thing for osmotic pressure. There are a lot of factors that can affect enzyme activity. Temperature being the best example that we can talk about in class, but it is not the only example. Something that can also affect the rate of reaction is substrate concentration. What's interesting is the rate of the product formation will depend upon how many active sites are available and how much substrate is present. So this is showing us, this top one, that there's a lot of extra enzymes and they are not full with substrate, meaning there's not a ton of substrate and there are still some enzymes that are just not being utilized. So it maxes out at a certain concentration. This smaller one, or the lower one, is indicating that there's more substrate than there are enzymes, which means you can't get more products faster because, well, the maximum rate is limited by the number of active sites. So there's only 6 active sites, and let's say there's, I think, 9, 10 substrates. Well, if there's only 6 active sites, only 6 of them will bind. And you'll have some substrates that just float around and don't do anything. So you can see that the maximum reaction rate is significantly less when you have a lower enzyme concentration. When you have a higher enzyme concentration, the maximum reaction rate increases. And that's just because there are enough active sites to meet the needs of all of those substrates. And you may have some enzymes left over. So this is a really great one that shows not only does temperature, pH, salt concentration, osmotic pressure, but also concentration of enzymes and concentration of substrates also affect the rate of reaction. So there are a lot of things that go into rates of reaction of enzymes. And we could do calculations and figure out specific rates, but we're not going to do that in this class. Competitive inhibition, again, we've talked about, but that's okay. Basically, competitive inhibition is when you have a inhibitor competing against the substrate, for the active site. So if you notice on this top one, this enzyme, there's more inhibitors than there are substrates. So the inhibitors are going to compete for the active site. And when they bind to the active site, the substrate can no longer enter. Therefore, you inhibit the enzymatic activity. If you have more substrate than you do inhibitors, then the chance is it's more likely that the enzyme does not become inhibited, right? You have 10 substrate molecules and 2 inhibitors. Well, there's a higher likelihood that the substrate will bind to the active site than the inhibitor itself. Now, these reactions are reversible, meaning it's not permanent. So if at one point you have too much of an inhibitor, you can actually switch that around so then you have more substrate than an inhibitor. So it's reversible. And this describes that down here. Basically, increasing substrate concentration can restore the rate. So competitive inhibition, inhibitor competes for the active site to slow down rates of reaction. Non-competitive inhibition, you have an inhibitor, a non-competitive inhibitor, that will bind to the allosteric site. When it binds to the allosteric site, it will prevent the substrate from being worked on. The substrate can still bind to the active site, possibly. Sometimes it can. Sometimes when the inhibitor binds to the allosteric site, the active site changes shape and the substrate won't be able to bind. But even if it does, having an inhibitor at the allosteric site stops the substrate from being worked on. But if we're missing an inhibitor at the allosteric site, then it's just normal enzymatic activity and you get your products. So non-competitive inhibitors, they don't compete for the active site like competitive inhibition. Instead, they find elsewhere to bind, the allosteric site, and it will prevent the substrate from actually being worked on. All right. Both ways of slowing down rates of reaction. Right, so this is what I was just talking about. The inhibitor binds to the allosteric site. Okay. Allosteric site. Now, we talked about it as an inhibitor. The allosteric site can also be an activator as well, but we're not going to focus on that. We're only going to focus on the fact that the allosteric site can be is a non-competitive inhibitor site. And then this just shows the allosteric inhibition versus what I just said with the activation. Don't worry about the bottom one here. This is the one we're focusing on. The fact that the inhibitor binds to the allosteric site and then it slows or stops the rate of the reaction. And then the middle one here is just normal. And then sometimes you have to have an activator in the allosteric site to get the enzyme to work or increase its functionality. But we're not focusing on activation. We're only focusing on inhibition. And again, this is reversible, right? So if all of a sudden this inhibitor releases, then it goes back to normal, like this one in the middle. All right. So now that we've talked about that, we're going to move on to identifying bacterial samples. So to identify an unknown sample, think about it clinically. You get a sample from a patient, throat swab, nasal swab, whatever it may be, and it comes down to the lab. You're looking at this swab, and you say, all right, let's plate it on some type of media. If you put it on something general like TSA or nutrient agar, do you think that sample is only going to have one bacterial species in it? You take a nasal swab from somebody, you culture it, you put it on a plate, and you say, all right, let's see what happens. Do you honestly think that there would be only one bacterial species there? And I hope all of you immediately said no, because when you take a nasal swab, throat swab, skin swab, doesn't even matter. You're going to actually take your fingers in lab 3 and rub them across the plate. So skin sample, bacteria on your fingers. They're not pure cultures. You have to get pure cultures. You don't start out with them though. So to get a pure culture, we use the streak plate method, which you've seen in lab. Streaking for isolation, streak plate method. So we also use differential and selective media, as well as other tests. We can use microscopy. We can stain them, gram staining, simple staining, endospore staining, flagella staining. There's lots of different types. And then what we can use is just general characteristics to start narrowing it down, and then getting into more specific tests. But we do need a pure culture to work with. Remember, Koch's postulates, right? We have to get a pure culture to prove that it's that organism causing the disease. Some of the ways we do that are catabolism tests. Basically, do they break down certain amino acids? This one right here is a good example of that. Or even fermentation tests. So this right here, A, actually no, excuse me, B, is a media that has specific components in it. Let's say it's got glucose in it. It also has a pH indicator. Also has a pH indicator telling it if the pH will change. And notice it also has an upside down test tube. So there's a big test tube, this really big one, and then inside is an upside down test tube that has been filled with this media. The media has kind of filled up into this upside down test tube. This upside down test tube has a special name. It's called a Durham tube. And basically what that is if the bacteria that's put into this this broth, let's say it's got a sugar in it and it's just this broth, if the bacteria in here can ferment the sugar, there will be a pH change, which is why you will get a color change, because there's an indicator, a chemical in there that says, hey, pH has changed, so it changes color. We've seen this before in MSA plates, mannitol salt agar plates, right? If there's it's differential, because if you see a change in color, that tells you something has happened and the pH has changed. In this case, it would be fermenting a sugar. The Durham tube tells us if the organism produces gas as a byproduct. You can see there's no bubble on the right, but on the left side, there is a bubble at the top of the Durham tube. What that does is that says, hey, guess what? This organism produces gas as a byproduct. And that's useful because not all organisms produce gaseous byproducts. So like CO2, for instance, is not produced by all organisms as a byproduct. It just happens to be with whatever this organism is. So, point being, we can use fermentation tests, we can use amino acid catabolism tests, we could use staining, we can use just microscopy, we can use a lot of different techniques to try and identify bacteria. The problem is some bacteria have really similar characteristics. So the more specific we can get with tests, the closer and quicker we can get to an identification. And another test we can use is the catalase test. Catalase, if you recall, ends in ase, so it's an enzyme. If you recall, when we talked about oxygen requirements, aerobic bacteria had catalase and superoxide dismutase. Both of them broke down atmospheric oxygen. Well, superoxide dismutase broke down atmospheric oxygen. Catalase then took the hydrogen peroxide that was formed and breaks that down. So if an organism is aerobic, it has both SOD and catalase. And in this case, you take hydrogen peroxide, you introduce the bacteria to a little bit of drop of peroxide on a slide, and if you get bubbles, that tells you that it is producing oxygen as a byproduct. So for instance, peroxide is H2O2, that's its chemical formula. When bacteria have catalase, catalase breaks H2O2 into H2O and O2. So you get oxygen as a byproduct and you see bubbles. That tells you have catalase. Not all organisms have catalase. Obligate anaerobes lack catalase. They also lack superoxide dismutase. So you can actually, from a simple test of hydrogen peroxide and whether it makes bubbles or not, you can say, all right, well, this is either a obligate aerobe or facultative anaerobe, but it can't be an obligate anaerobe because obligate anaerobes die in the presence of oxygen and they lack catalase. So you can actually get rid of a whole bunch of organisms right off the bat. And then on top of that, using the catalase test, we can narrow it down even further. This is a really neat tool that I use from a lab manual that was produced by a professor that I worked with when I was in graduate school. And I taught this exact same course in graduate school. In his lab manual, we used this. He built this. And basically, let's say you get a sample, you gram stain it, you look under a microscope, and you see gram-positive cocci. There are a lot of organisms that are gram-positive cocci. Okay? Arrangement can help, right? If it's streptococci, then it's in chains. If it's staphylococci, it's in clusters. But what if you have a very thick sample and you can't really tell? It looks like you have some clusters, you have some chains. You say, all right, well, this is not a really good way to go about, identifying. But in a very fast manner, you can determine whether or not you have staph or strep just by running the catalase test. Because staphylococci are catalase positive, which would mean they would produce bubbles, they would break down hydrogen peroxide. And streptococci are catalase negative, meaning they don't have the ability to break down hydrogen peroxide. So immediately, just by simply taking a small sample, adding it to catalase, you can say, all right, well, that's pretty impressive. I'm pretty sure I have staphylococci if it's catalase positive, or I don't have any bubbles, therefore it might be streptococci. Now, this is not perfect, but it is very, very, very useful in rapid identification. And then from there, you can look for other traits. So if it's catalase positive, we say, all right, we think it's staphylococci then. Where can we go from there? Well, if we think it's staphylococci, or staphylococcus, excuse me, we can use blood agar. And if we use blood agar and we see gamma hemolysis or gamma hemolytic colonies, meaning there was no red blood cell breaking down. It could be one of these two species. Or if we saw beta hemolysis, meaning complete breakdown of red blood cells, it could be Staph aureus. But we could even take it a step further and say, all right, well, I have a beta hemolytic colony. There are quite a few organisms that are beta hemolytic. But now let's test coagulase. Coagulase is an enzyme that Staph aureus produces. It's the only staphylococcus that produces coagulase. And coagulase basically takes the plasma portion of blood and clots it. Now, the plasma portion of blood is antibodies, water, dissolved substances, pretty much everything except for red blood cells and white blood cells. Platelets, for instance. So there's all that mixture, all that stuff except for the red blood cells and the white blood cells. And Staph aureus is coagulase positive. So you line up 4 test tubes, you take your sample, you take a colony, and you put it into test tube 1. You take another colony, put it into test tube 2, 3, 4. All these test tubes have plasma in them. You let it grow for 16 hours. You let it sit there for 16 hours. You come back, and you go to test tube 1, and you wiggle it around a little bit, and it's still liquid. Okay, that's coagulase negative. Then the next one, you wiggle it, it's still coagulase negative. And then the third one, you wiggle it, it doesn't move. It looks solid. You can literally flip the test tube upside down and nothing will come out because the coagulase will turn it into almost this hardened substance in the test tube that just, it won't come out. It won't come out because the coagulase forced all the plasma to clot. So that is a confirmatory test. That is an absolute confirmatory test. So basically, you get beta hemolytic colonies, and you're like, well, I'm pretty sure it's Staph aureus, but let me use a confirmatory test like the coagulase test to be sure. And if you get a positive result, then you are sure it is Staph aureus. Now, there are other tests you can run to double and triple check, but Staph aureus is the only Staphylococcus species that is coagulase positive. All right, again, this is all really useful for rapid identification. We've already talked about this. Sorry, this slide should actually not be here. That was earlier. We saw this. This should not be here. Sorry. Another way to test is what we call the API system, or what we call in the enterotube. The technical name is the API system, but we can call it what we call the enterotube. It is a tube that has about 15 different tests in each of its little compartments. So for each compartment, there is a different test. Okay, each compartment, there's a different test. And basically what you do is you take a sample on the end of like a metal rod. There's a metal rod that runs behind all these tests. And you remove the end and you pick up sample on the rod that's right here. And then you pull the rod and you pull it out the other end. And as you pull from here with the sample, it inoculates every single one of these little chambers as you pull it through. You then put this in an incubator for 24 hours or so, and then you come back and you look at each individual test. So all of these indicate it's testing for something. So this one's testing for citrate. This one's testing for glucose. This one's for mannitol. This one is for amylase. So it's testing for a whole bunch of different things at once. This is a very, very useful tool, and it is one of the best ways to identify unknowns, because each organism is going to have a distinct reaction or non-reaction to all of these tests. So there are codes. We have a book called we have a book that goes with this. It's, gosh, thicker than your textbook. And basically, we look at all the tests and we determine positive or negative, and then each one has a value associated with it. So for instance, we have a positive, negative, and a positive. So we take the two positives, we add them together, there's our first digit. Then the next three. 1, 2, 3, we only have one positive, and that value is 2. So we get a 2. And then so on and so forth. And eventually you get a 7-digit code. You open the book, you go to the number, it's numeric, the list is in numeric order. You go to 5215773 and figure out that it is Klebsiella pneumonia. Now, there is a problem with this. It's not a huge problem, but it is a drawback. Interpretation of a positive versus a negative can be subjective. The way you see it, you say, well, maybe there was a slight color change in this one. Maybe, maybe not. I'm not sure. It looks close. That's a problem. I may look at it and say, yeah, that's a color change. And you may say, well, I'm looking at it and it looks kind of maybe possibly a color change, but possibly not. So then it kind of messes up with your code a little bit, and you actually have to calculate it both ways. And that's only assuming you disagree on one of them. Let's say you disagreed on like 5 of these tests, these individual compartments. Let's say you disagreed on 5 of them with me. Then all hell breaks loose, because then we really can't calculate this number. So it's great. It is honestly one of the best tools to use. We don't use it in our class only because they're very, 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 very expensive. Just one of these is about $500 for just one. So they're pretty expensive. But the amount of information you get is extremely valuable. And even if you don't get an exact organism, it at least points you in the right direction. So in terms of testing, using staining and microscopy, those are not the best. They're meh. Using biochemical testing is better. And then using antibody testing is even better than that. So when you test for a bacteria and you say, I think it's this one, and you look at it under a microscope and you gram stain it, unfortunately you can't tell. I mean, there are so many different species that are gram-positive cocci or gram-negative rods that it's just a terrible way to try and identify an organism. It's helpful in that it gives you an idea of what the arrangement morphology is and the gram reaction, but it doesn't really do that much for identification. It can help you point you in a direction, but not necessarily give you the specific answer. All right. So the best way to test is using one of these API systems, an enterotube, and or using antibody testing, or what we call molecular testing. So ELISA, for instance, is a really good one. We haven't talked about ELISA in this class, but molecular testing is really useful and it's very powerful. So I suggest, if I were you, to look up what ELISA is, ELIZA, all caps, ELISA, and just take a look at it. It's a really powerful tool. Now this slide is just showing you a summary. Here's metabolism, ATP, here are the reactions, etc. This is just a summary slide. Same thing here, this is a summary slide of enzymatic activity, and then some biochemical tests. Biochemical tests are great. Okay, so that's the summary for that. We have a little bit more to go through though. So I want to revisit genetics a little bit and talk about gene transfer. Now, granted, this semester being the way it's set up, you all are not identifying an unknown bacteria, but I promise you, it gets about this crazy when you try to identify an unknown species. It's very, very, very challenging. Anyway, point being, I want to revisit genetics and gene transfer just a little bit. So the central dogma we know is DNA, RNA, proteins. But there are two ways we consider genetic information to be moved around and passed on. There's what we call horizontal gene transfer and vertical gene transfer. Horizontal are these two right here, expression and recombination. And then vertical is this method right here, which is just replication. So when I say an example of horizontal gene transfer is expression, meaning you get protein expression. Transcription, translation, and the protein is functional. I could also say recombination. Basically, the cells can transfer sections of DNA to other cells of the same generation. So this recombinant cell right here, this purple section is its original DNA. This right here, this lighter colored section, it got from another cell. And we're going to talk about ways that recombination can occur. So it got this extra DNA right here from somewhere else. And then if I say vertical gene transfer, I'm simply talking about binary fission. One cell copying the genetic information to make two identical daughter cells. Right here. Okay? So, horizontal gene transfer, expression and recombination. Vertical gene transfer, replication. Binary fission. Alright. So, this image right here, I'm not spending a lot of time on. If you want to pause it and look at it, great. But I spent a whole hour-long video talking about DNA replication and transcription translation. So this is just a video from another book, not your current book, but a book I used to use that you may want to take a peek at. Same thing here. This is just showing you replication, binary fission of the cell first replicating the DNA. Okay, so this is vertical gene transfer, right? Because it's copying the DNA to send one copy to one new cell, one copy to the other new cell. All right, so that was vertical gene transfer. I spent a ton of time on that before. I'm really not going to focus on it any more than I have to now. I want to talk about recombination. So this is a chromosome map of E. coli, And notice how huge, look at the chromosome. Look at all that genetic material. That's huge. In comparison to the size of the bacterial cell, right? It looks really big. This shows us the chromosome, the whole size, the map, and then it tells us locations of certain genes. So right here, all these genes are grouped together, are associated with membrane synthesis. All the ones right here are carbohydrate metabolism. All the ones here are DNA replication and repair. Remember, bacterial cells group genes together in blank. They group them together in operons, where they get transcribed and translated together. So all of these are going to get transcribed and translated together. Now, in addition to the chromosomes, some bacteria have what we call plasmids. Plasmids are extra pieces of genetic material that they've picked up from somewhere else. So this small circle right here is a plasmid. This is the chromosome. So this is the bacterial chromosome, all its genetic information right there. But it can have a plasmid which has other genes that are not included in the chromosome. And it can pick up plasmids multiple ways. They're really small plasmids, very, 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 very small. They only carry a few genes. And some of the genes are useful, some of them are useless. So this plasmid may or may not be useful to this cell. Not every one of them is going to be revolutionary and amazing. It may just be something that, man, the cell doesn't need or want. It's just there. So here, my point, occasionally carry important genes. And if we were to highlight something specific, we would say antibiotic resistance genes. That's how some bacteria become antibiotic resistant, is they get plasmids that have the resistant genes on them. So when expression occurs, the plasmid expresses those genes, and therefore the cell becomes resistant. So resistance factors, plasmids can be transferred vertically or horizontally. So when you go through vertical gene transfer, copies the chromosome, also the plasmid gets copied as well. Plasmids have their own origin of replication, meaning they copy themselves. So when the cell goes into vertical gene transfer, this chromosome gets copied, and the plasmid copies itself, so the two daughter cells have not only the chromosomes, but the plasmid as well. So one cell would have the chromosome and the plasmid, the other cell would also have the chromosome and the plasmid. So they can be transferred that way, vertically. Horizontally, they can also be transferred between cells of the same generation. So again, vertical gene transfer is here we have replication, two identical daughter cells, and then here's horizontal gene transfer with recombination, swapping genes between cells of the same generation. Yep, so vertical, reproduction of generation of cells. Horizontal, transfer genes between cells of the same generation. Perfect. I've kind of said this multiple times already. So 3 modes of horizontal gene transfer in bacteria. We're going to talk about all 3. Transduction, where DNA is carried by a virus. Transformation, where DNA is transferred in a solution. And conjugation, where DNA is transferred through a sex pilus, a bridge between 2 cells. Conjugation, honestly, is pretty close to bacterial. It's close enough to what we call bacterial sex, essentially. A bridge forms between one cell to the other, and it exchanges genetic material. pretty much bacterial sex. We'll look at each one of these. So first, this is recombination. A phage, which is what we call bacteriophage, is what infects bacteria. So bacteria have their own special viruses that infect them. So bacteriophages are viruses that infect bacterial cells. And you can see here the phage sends its DNA in, the DNA chops up the host genome, more viruses are made, but instead of making all viruses with only phage DNA, sometimes there are accidents where the DNA from the original bacterial chromosome gets packaged instead. And when the viruses leave and infect other cells, instead of infecting it with viral genetic material, it actually infects it with bacterial genetic material. And then the bacterial genetic material can get incorporated into the host genome. So, on accident, viruses, phages can accidentally transfer bacterial DNA from one cell to another instead of viral DNA. It's very interesting in the way it works. I have a video, or I have links to videos on the last slide in this chapter that I would encourage you all to watch to help understand these a little bit better. So transduction by a bacteriophage, infect the bacteria, breaks down its chromosome, makes new viruses. Sometimes the DNA that's been broken down in the bacterial chromosome gets incorporated into the virus instead of viral DNA, and they accidentally transmit that to a new host. Easy enough. Transformation is when you have free-flowing or free-floating DNA fragments, typically in a solution. So here's your competent cell. Here's the DNA fragments. It will enter into the cell. It'll take the DNA and enter into the cell, and then it'll align itself properly with the correct base pairs. So it'll align itself so it matches. It will then cut a little hole and insert the DNA, the donor DNA, into the recipient DNA. So this chromosome has now got new genetic material incorporated into it. And then whatever's left over gets broken down. And you can see right here, you have a small section that has now been incorporated into the genome. Now it may carry some really useful genes. It doesn't necessarily have to, but it can. We do this in the lab, not in BISC 210, but in research labs. We take E. coli, and if we wanted to express a certain gene, let's say we wanted to do something like make a certain protein, we can put the gene in solution, the genetic material, the genes in solution, put the E. coli cells in there, and then kind of trigger the cells to take the DNA up into the cell and then incorporate it into its chromosome. So we can actually do this in the lab and try to get sections of desired genetic material incorporated into the host genome. It's actually a fun thing to do. One of the ways you can do it chemically by weakening the cell wall, or you can do it basically through vibrations. You can have certain frequencies that kind of vibrate and trigger the cell wall to get little holes in it, and then the DNA can just float right into the cell using those new openings. It's really neat. And then the last one is conjugation. Basically, Conjugation allows for a cell that has the F plasmid to create a bridge and then send the F plasmid to a new cell. And the same thing here, showing you the bridge forms. It's copying the F plasmid, sending it into this cell. And then at the very end, now you have two cells that both have the F plus plasmid, meaning it's called the fertility plasmid. That's why it's the F plasmid, fertility plasmid. F plus means it has it, F minus means it does not. If a cell is F plus, then it has the fertility plasmid and can form this mating bridge. And that allows for cells to transfer genetic material as well. Okay, you can end up with recombination and transferring material between cells as well. Conjugation is very, very powerful. So, there's short videos that I have links to right here that you should watch. There's 3 of them, 1, 2, 3, each on a different way of recombination. So conjugation, transduction, transformation. Watch them, pay close attention, because they are really useful. All right, that is the end of this chapter, chapter 8. There will be some participation questions going up shortly. Hopefully that'll help clear up any confusion. And then I have office hours if you want to ask me questions about Chapter 8 at all, Wednesday and Thursday from 12 to 2. All right, have a good day, and I will talk to you all soon.